Hey Team Bio, today you are going to learn how to solve a murder mystery. This is going to be a screencast on DNA fingerprinting. I'm sure in maybe movies or TV shows or in the paper you've read about people, about crimes being solved using DNA evidence, and today you're going to learn about the process of how that actually works. Now, this is important, obviously, because it helps us solve murders or crimes, but it's also pretty complicated. So saddle up, get ready for a potentially long and confusing process. Happy long weekend. No, it won't be that bad. Okay, so DNA fingerprinting, broken down into two different components. There is PCR and gel electrophoresis. And I'm going to talk about the first part first and gel electrophoresis second. Okay, so PCR. PCR stands for polymerase chain reaction. Write that down, P-O-L. Polymerase chain reaction. And so you might have heard some of these words before, especially the word polymerase. What does that mean? Or to polymerize something? It's to make a many-mer. The mer that we're making many of is DNA. So at a crime scene, let's say this is the victim's shirt. Potentially there's victim blood all over it, but also let's say the killer left a blood stain or even maybe left a piece of hair with a hair follicle at the end of it, which has cells that have DNA. Well, the police are going to collect these cells, either a blood sample or a hair follicle, and they are going to take that DNA, but the problem is that there isn't very much of it. So they need to figure out a way to make more of this DNA. So polymerase chain reaction was invented by this guy named Kary Mullis. And he may or may not have been high on acid when he invented this technique. And then it went on to win him a Nobel Prize. So I don't know what the moral of that story is. I'll let you decide. Don't do drugs, but polymerase chain reaction is a pretty powerful technique that you're going to learn about right now. Okay, so in a blood sample, even though there are many cells in a blood sample or in a hair follicle, there's still, and a lot of DNA in those cells, there's still not enough. So I'm just going to say, even though we have a lot of DNA, I'm just going to pretend like we only have one strand. Okay, so there's one strand of DNA right here, and we want to make more. So this is what we do. First, add heat. This is going to cause the DNA to denature, much like we studied earlier. Proteins denature when you heat them up because the hydrogen bonds that hold the structure together, the secondary, tertiary, quaternary structure together, break. Well, the same thing with DNA. The hydrogen bonds that hold complementary base pairs together break apart when you heat it up, and you get denaturation. And then you're going to add primers to your DNA, and they're going to anneal, so RNA primers to the DNA. And then you can add a bunch of free nucleotides to the solution. And then you add a DNA polymerase. So add DNA polymerase, and DNA polymerase will synthesize down the length of the strand, and voila! At the end of one round of replication, you have 2 full pieces of DNA. Now, you do the same thing. You add heat, and the process repeats itself. You have denaturation, of each strand. The primers, which I should have drawn over here as well. The primers are just hanging out in the test tube solution with the nucleotides. The primers anneal to the DNA on either side. Oh, this side and this side. And the DNA polymerase, zips down each side, and you get polymerization. So now you end up with 4 pieces of DNA. Okay, so you guys might have caught on to something here, that you heated the DNA in between after you polymerized, and so why didn't these DNA polymerases right here denature? Well, the answer to that is that this is a special kind of polymerase called Taq polymerase. It's taken from a bacterium that lives in hydrothermal vents, really areas where water can be boiling or almost boiling. And so this polymerase, when heated up to the temperature that denatures the DNA, the polymerase actually doesn't denature. So it can survive rounds of heating and cooling. And so while the DNA separates and denatures, the Taq polymerase doesn't. So as long as you start with Taq polymerase, primers, free nucleotides, and some pieces of DNA, ultimately you'll end up with a ton of DNA. So you could have started from one cell, or probably hundreds of cells, and end up with millions of pieces of DNA at the end of multiple rounds of PCR. Okay, so that's the first step of DNA fingerprinting. PCR results in DNA, not NDA, DNA. DNA amplification. Okay, the second round, gel electrophoresis. In this round, we are going to separate the DNA and analyze it. Okay, so we just did our PCR. Let's go back to our crime scene here. We have our victim's shirt. It's covered with the victim's blood. Let's say there was a stab wound to the chest. So victim's blood over here, but the victim also struggled, and we think we have a sample of our suspect blood right here. Okay, so what do? Well, we run, I just said blue. This is blood. Okay, so we run PCR on both these things, the suspect's blood or suspect blood, the victim's blood taken from the shirt, and maybe also victim's blood taken from the body. And we end up with a bunch of different samples. So, okay, so I'm going to talk you through this crime scene. Okay, so we've got our bloody shirt. We've got the victim's blood on the shirt. We've got one of our suspect's blood, or a suspect blood on the shirt. Whoever killed this person, we think their blood is on the shirt. Okay, so we take a sample of the victim's blood from the body, and we run PCR. We take the victim's blood from the shirt, so this sample right here, and we run PCR on it. And we take the crime scene suspect blood, so this, and run PCR on it as well. And then we have a few suspects in the case. Let's say we have 3. Suspect 1, Suspect 2, Suspect 3. We take DNA samples from each of them, so we ask them to give a blood sample. And then we run PCR on all 3 of those as well. So now we have a ton of DNA. And what we're going to do is we're going to take that DNA and we're going to expose them each independently to these enzymes called restriction enzymes. Okay. Okay, so restriction enzymes are enzymes that cut DNA, and they cut DNA at specific sequences. So for example, there's a restriction enzyme called HindD3, and it cuts DNA whenever it encounters this specific sequence, A, G, C, T, T. That's on one side. On the other side, it would be T, T, C, G, A. Now, it cuts it just like this, right down between the two base pairs, like so. You might notice that this is a DNA palindrome. It reads A, G, C, T, T in this direction, and then A, G, C, T, T in that direction on the underside. Have you ever heard of palindromes like race car is a palindrome? It says the same thing forwards and backwards. Well, DNA palindrome is the 5 to 3 prime side in one direction reads the same as the 5 to 3 prime in the other direction. And interestingly enough, restriction enzymes often cut at these palindromes. Not all restriction enzymes cut at palindromes, but many do. So, anyway, you have your HindD3. You're going to throw it in with the DNA, and if this restriction enzyme encounters this sequence, it's going to cut. So let's imagine in this DNA we find this sequence right spot in the middle, like so. Well, you're going to end up with two pieces of DNA that are exactly the same size. It has these little funny things called sticky ends that overhang. We're not going to talk about them now, but what we can talk, well, I don't actually know if we're going to talk about them later. Anyway, all you need to know for our class is that you end up with two pieces of DNA, in this case of the same size. So this is potentially one person. Let's say this is me. This is my DNA, and we cut it this way, and we end up with two pieces of the same size. Well, let's say we take you and expose you to the same restriction enzyme. Well, because all people are slightly different, have slightly different DNA sequences, you might have the HindD3 site over here. And so instead of two equal-sized pieces of DNA, you get one short little piece of DNA and one long piece of DNA. The differences, or these little fragments, we call RFLPs, RFLPs, or restriction fragment length polymorphism. I'm going to write that out. Okay, restriction fragment length polymorphism. What that means is that we have a restriction fragment. We call it that because we cut it with a restriction enzyme. And a length polymorphism, or a polymorphism length, would be different lengths. So we end up with different length pieces. So a polymorphism is just two or more phenotypes within a population. So for example, brown eyes, blue eyes, green eyes, black eyes, those are different polymorphisms of the same gene. We call this a polymorphism because we have different fragment lengths within people. Okay, but you should just know these as RFLPs, RFLPs, and it's important that they can be different sizes. Okay, so let's go back to our setup here. I'm going to copy this and put it on a new page. Okay, so we have all these different people and all their DNA, and we're basically just going to do this process. We're going to take their DNA and expose it to these restriction enzymes, and it's going to cut it up so that they're all different sizes. But here's the deal. If the DNA is just in a test tube, like so, let's say we have all our different samples of DNA in a test tube, and we cut it all up, and so we have all these different sized fragments. Everyone's got their own personal RFLPs in here. Well, that doesn't do us much good because we can't see it. So we need a way to separate these pieces of DNA by size, because it's these different sizes here that are going to tell us something about who committed the crime. We're looking for a match between our crime scene suspect blood and either our suspect 1, 2, or 3, or maybe none of them will match and we'll rule them all out. But we need to look for the same size restriction fragment length polymorphisms in here and compare them to what these RIFLIPs looks like. So here's how we separate the DNA out by size. We're not going to take these test tubes. We're going to delete that. We do something, it's called running this out on a gel. So I'm going to draw you a gel. The gel is made up of this stuff called agarose. And you can think of it as kind of like Jell-O. So it's like a little brick of Jell-O. And in this little brick of Jell-O, okay, there are little divots in this brick of Jell-O here, which are called wells. And what we do is we take our DNA sample, our multiple DNA samples. So I've only drawn 3 wells here, but in a gel with all these different suspects and samples, we would actually need 6 wells. But we fill these wells up with our DNA that has just been exposed to our restriction enzyme, so it's been all cut up. So these wells are all filled with different DNA. And we do what's called running the gel. So now I've shown it to you in 3D. I'm going to show it to you now from top down. We take the gel with our little wells in it, right here, and we put it in a contraption that has a buffer solution, so like a salt water solution here, and it's connected to electricity. So we plug it into the wall right here, and we put the positive electrode right here and the negative electrode up here. Now this is, remember, DNA samples in here, and DNA is a negatively charged molecule. So it's going to run down the gels towards the positively charged side. But remember, it's running through this agarose gel. And the agarose gel is, it would be like you trying to run through Jell-O. It's difficult to get through. There are all these little pieces that the DNA has to run through. And I want you to imagine if you were running through a gel by yourself. You could kind of dodge and duck and weave through. You would be slowed down. It wouldn't be as fast as just running on a field, but you could go pretty far. Now imagine you were to do the same thing, but I asked you to hold hands with everyone in our class as you were running through the gel. Well, that would probably take you a little bit longer. You're going to get stuck on little pieces of the gel and have to kind of wend your way through. Well, that's what happens with DNA. The small pieces of DNA migrate through the gel faster than the large pieces of DNA. So you end up getting separation of the DNA by size. So if we take this gel, I'm going to erase all these lines that I've drawn on it. We plug it into the wall, we run a current through it, the DNA starts to migrate down the gel, and ultimately we start to get a banding pattern. Maybe looks like this for this one. And this for that one. Okay, you'll notice that so again, these are our small fragments of DNA here, and these are our large fragments of DNA, and they come from our different RIFLIPs here. So This would be a medium-sized piece of DNA. These pieces would actually migrate in the same spot in the gel. This would go far, this would not go very far. Okay, so back to our gel. These are our small pieces, these are our large pieces. And let's say that this is the crime scene DNA taken from the victim's shirt. So this is we think it's our killer. This is, we can say, suspect 1, suspect 2, and suspect 3. Well, you notice we can automatically rule out suspect 1. They do not have the same banding pattern, the same riff lips, as the DNA taken from the crime scene. So they are not our killer. But suspect 2 and suspect 3 have the same riff lip pattern. Well, who did it? Well, this is what we can do now. We can take a thing called a probe. Probes are often radioactive. And they only bind to certain sequences of DNA. So a probe would be a short section of RNA that has a certain sequence, and it only binds to the complementary sequence. So we can take our probe and we can throw it in our gel electrophoresis, and wherever it has complementary sequence binding, it's going to anneal. So remember, all these people, while they might have the same size fragments, they don't have necessarily the same pieces of DNA within those fragments. So right now we're separating DNA out by size, and now we're probing to see what pieces of DNA are where on the gel. So let's say our crime scene DNA has a probes anneal here and here, and our suspect 2 has a probe that anneals here, and our suspect 3 has probes that anneal here and here. Well, we found our killer. Suspect 2 can be ruled out because of the probe, and suspect 3, because they have the same riff lip pattern and the same probe binding pattern, is our killer. So, this was really long. I'm sorry, I'm going to end it right now. See you in class on Monday. But at least now you know how to solve a mystery through DNA, PCR, and gel electrophoresis. Okay, bye.